Morning to all. I, Mr. Prashanth Pai M, welcome all the participants to the awareness webinar on Can You Change Your Destiny? A Glance Beyond Genetics, which is organized by Sanatana Samprathishtana, Mysore, in association with the Rotary Club of Mysore-West. Prayer is the process of interacting with God in order to achieve a purpose. So let us begin this webinar by seeking the blessing of the Lord Almighty. Namaste Sharada Devi, Kashmira Puravasini, Tvamaham Prarthaye Nityam, Vidyadanam Cha Dehime. Now, I request Dr. Ramachandra C G, the Managing Trustee of SANATHANA SAMPRATHISHTANA, to welcome and introduce the speaker of today's webinar. I also request him to welcome the guest and the participants. Over to you, sir.
Very good morning, one and all. I take this opportunity on behalf of Sanatana Samprathishtana, Mysuru, to welcome today's speaker, Dr. Shruthi Kanthaje, today's guest, Dr. Raghavendra Prasad M D, President, and Shri Dinesh Kumar, Secretary, Rotary Club of Mysore-West, Shri Lokesh C G, President, Dr. Shantharaje Urs and Smt Bhagya Gopal, Board of Trustees, Sanatana Samprathishtana, and finally, all the participants. So Sanatana Samprathishtana Trust is a non-profit organization situated at Mysuru, Karnataka, has been serving the society and supporting noble causes since the time of its inception, and through the process of its evolution. The sole essence of Sanatana Samprathishtana Trust has always been for the benefit of the society, to guide, to help, to educate, to empower, and to enrich the knowledge of any individual or group of people. The Trust provides a broad range of social services to all the categories of people. So these services are available regardless of age, religious preference, gender, economic status, etc. Some of the programs that we run and even proposed programs under the Sanatana Samprathishtana Trust are Research and Training Institute, Skill Development Center, Coaching Center, Employment Center, Sports Academy, Cultural Academy, Organics and Herbal Ayurvedic Health Care Center, Meditation and Yoga Center, and many more. And coming to today's speaker, let me introduce today's speaker, Dr. Shruthi Kanthaje. She has completed her BE in Biotechnology from VTU Belagavi in the year 2009 and MTech in Biotechnology from Padma Shri Dr. D.Y. Patil University, Mumbai in the year 2012. She has received her PhD in Cancer Biology in the year 2018 from PGI MER Chandigarh for the thesis titled Role of Deregulated MicroRNAs in Sorafenib Resistance and Metastasis of Hepatocellular Carcinoma. So currently she is working as Post-Doctoral Fellow at Yenepoya Research Centre, Yenepoya-Deemed to be University, Mangaluru, Karnataka. She has exposed to several practical and theoretical aspects of microbiology, biochemistry, molecular biology, immunology, structural biology, and bioinformatics. Her research primarily involves understanding the potential of biomolecules like microRNAs, significantly altered signaling pathways to improve diagnostics, therapeutics, and provide better clinical management of cancer patients. She has qualified GATE with a percentile of 92.5 in the year 2012, qualified GRE with a score of 310 in 2013, also secured third rank in PhD entrance exam PGI MER Chandigarh in the year 2013. She has worked as a trainee under Biotech Industrial Training Program sponsored by DBT at Biological E Limited, Hyderabad, as a research training and junior research fellow at ACGREC, Mumbai, as a junior research fellow from PGI MER Chandigarh, as a senior research fellow from ICMR at PGI MER Chandigarh. She has published many papers in reputed national and international journals. She has won first place in poster presentation at 7th International Conference and Expo on Molecular and Cancer Biomakers, Berlin, Germany. She has participated and presented papers like posters in various national and international conferences and consistently bagged the first place. She is trained in Carnatic music and Bharatanatyam from more than 10 years. So with this, the brief, the introduction of today's speaker, once again, I'll welcome to share the knowledge with us. So the basic intention to organize today's awareness webinar on can you change your destiny? A glance beyond genetics. So it is all about understanding our own human body, its complexity at a molecular level. Here, speaker will introduce the vast topics such as genetics, DNA, genes, and of course, the topic of the day, epigenetics, in a comprehensible manner. Epigenetics here, epi means up, above, or beyond, and genetics means the study of heredity or the DNA sequence. Hence, epigenetics is the layer of information beyond genetics. It is the master controller of life, not making it too technical. She tried to give insights into the ingrowing world of epigenetics with interesting day-to-day examples. With this, I wish all the participants will have a fruitful interaction with the speaker. Once again, thank you, and all. Over to you, Prashanth.
Thank you, sir. Now I request Dr. Shruthi Kanthaje, Post-Doctoral Fellow, Yenepoya Research Centre, Yenepoya-Deemed to be University, Mangaluru, to start her session. Over to you, madam.
Hello, good morning, everybody. Am I audible?
Yes, you are audible, madam.
Yes.
Start the video as well. And, is my screen visible?
Yes, yes.
Yes, yes.
Visible.
Oh. A very good morning to all of you. I thank Prashanth Pai sir and Dr. Ramachandra for giving me this wonderful opportunity to be a part of this webinar. So the topic for today. So before starting the topic, as a speaker, for anybody, it's always exciting to speak offline on a live mode because you can see the audience live. And you can see, like, if somebody is yawning or somebody is looking at their mobile on and off, you can see you're not doing it very well. So you can change your tone and you can get the audience back to the talk. But unfortunately, it's an online talk, so I can't see all of you. But I would love this to be an interactive session. So if you have any doubts, you can disturb me anytime. You can stop my talk and unmute yourself and answer raise your questions. Or if you think you have something to add on, you can comment or raise and add on the points so that we all will benefit. So with this, I would like to start my talk. The topic for today is, can you change your destiny? A glance beyond genetics. So the first part of the talk, that is, can you change your destiny? So it looks like it's a motivational line, but believe me, I'm not a motivational speaker. I'm just a researcher. This is not going to be a motivational talk, but I would love if it turns out to be one. So that part you understand. The second part of the talk, a glance beyond genetics. Now here we have a word called genetics. If you're from a non-biology background, this might be a little alien to you. May I ask the audience, like, if you, what comes to your mind when you hear the word genetics? Can anybody answer me?
Genes, DNA, RNA, etc.
Correct. Exactly. So genetics means heredity, DNA, RNA, and inheritance. So genetics is the study of heredity, as Dr. Ramachandra sir rightly said. So, but here in this talk, we are going to learn something beyond genetics. So the answer to the question, can you change your destiny? I'm not going to answer it now. Rather, I'll put the question at the end of the session so that you can answer. I'm going to give you pure scientific facts, like how and why you can change your destiny. So to understand something beyond genetics, we need to first understand genetics. What is genetics, right? So, but to understand genetics, genetics is something inside us, something which is passed on from generation to generation. So that's something inside us. So to understand genetics, we need to understand the human body. So I'm going to start my talk with the human body. So the picture here on my slide is from a very blockbuster movie of 1974, Bhakta Kumbara. The man here, he's the one who lies in the hearts of every Kannadigas, Dr. Rajkumar sir. So can anybody from the audience tell me which song is this? If you are a Kannadiga, you must know this. Sorry? Manava, correct. Right. So this is Manava Dehavu Mule Mamsada Tadike. It's a very famous song, and it means, for the non-Kannadigas, I'm telling you, that it means that human body is nothing but made of organs. So it's not lyrics, it's for science, right? So human body is nothing but, it's made up of organs, different, which are performing their specific functions. So every organ performs its own function so that the body as on a whole can deliver its function, right? So you have the heart, which is pumping blood, you have the stomach, which is for digestion, you have the lungs, which are breathing, you have the liver, which is metabolizing, and so on. You have many organs. So I'm told many of you are from a background like our faculties or students of different colleges. So you can imagine the body as an institute or a college or an organization. So as an institute is made up of different departments, like you have the IT, you have the BP, you have the mechanical, you have HR, you have finance. So you need all of these to be working in collaboration. So if any one of this goes wrong, the institute suffers. Likewise, you have the body, which is made up of organs, and every organ must function well for the body to function on a whole. So you have the body, which is made up of organs. Now, what these organs are made up of? Organs are made up of tissues. It's a second level of organization. So, for example, if you take the heart, so many of us don't imagine heart to be like this. Instead, we remember heart only to be like this, but sorry to burst the bubble, it's not as cute as it looks like this. Heart looks somewhat like this. It's a schematic representation. Actually, it's much more uglier than this, with the blood, with slime, etc. But jokes apart, taking heart as an organ example, it is made up of different types of tissues. So it can be a muscle tissue, nerve tissue, blood tissue, etc. So every tissue is performing its own function so that the organ can perform its specific function. So when we compare our body to an institution, you have the departments, which are organs. Okay. So again, the department is made up of different types of people. You have the teaching staff, you have the non-teaching staff, you have the students. So many functional units making the department work as a unit, right? So likewise, you have tissues. Now, what are these tissues made up of? Tissues are a collection of cells. Again, next lower level of organization. It can be a similar group of cells or a different group of cells. So tissues are made up of functional units called cells. So if you take an organ to be a department, you have tissues are the functional units, like you have the teaching staff, which is a tissue, which are made up of different types of faculty, right? For each subject, you have different faculty. So likewise, in tissue, you have different types of cells delivering their action so that the tissue can perform as a whole, right? So from tissue, we come down to cells. Now, cell is something which is the lowest level of organization or the lowest functional unit of the whole body. So it can function independently by itself. Now, if you think this is the lowest we can go, we are wrong. Now, cell by itself is an independent body. You know, it's like human body. Like human body has organs, cell has organelles. Now, as you can see in the picture, can you see my cursor?
Yes, we can see.
Can anybody answer? Can you see my cursor?
Yes.
Okay. Okay. So cell is made up of organelles. So you have the nucleus, you have the mitochondrial reticulum, Golgi bodies, and so on. So many different types of organelles. Now, if you think this is the lowest we can, what are these organelles made up of? So organelles are made up of what we eat. You know, you have proteins. You might have heard about proteins, carbohydrates, lipids, you know, nucleic acids, all these. So the organelles are made up of different compositions, which are made up of proteins, lipids, carbohydrates, etc. Now, the proteins, again, if you want to go still lower, the proteins or these biomolecules, collectively, these are called biomolecules. And these proteins or nucleic acids, these are again made up of different monomers. They are like biopolymers made up of different units of monomers. For example, proteins are made up of amino acids, and you have nucleic acids, which are made up of nucleotides. Likewise, you have different biomolecules made up of different types of monomers. Now, what are these monomers made up of? You have the amino acids or the nucleotides, which I just spoke about. These are made up of, if you can remember, some of the elements, you know, if you have left chemistry on 12th, that's the last time you might have seen this kind of table. It's a periodic table, right? So only chemistry people follow it later and we stop it at there. But these biomolecules and the monomers of these biomolecules of some of the elements on these present in this period. And the human body is made up of some of these elements present on the periodic table. So it has to be 65% oxygen. It has to be, we breathe oxygen all the time, right? So 65% of oxygen, 18% carbon, 10% hydrogen, nitrogen, and trace some other metals, some other elements like calcium, phosphorus, magnesium. It all came down to these elements. But why were we go these why were we go genetic and inside the nucleus genetics structure of nucleus, we don't need to understand the structure of nucleus, but you can see some scribbled structure inside the nucleus here, very much related to genetics. Nucleic acid, there are two types of nucleic acids. One is DNA, RNA, as Prashanth Pai sir rightly said in the beginning when we asked about genetics. So one is DNA and another one is RNA. Both are in a subject itself. So today, we'll omit RNA, we'll not learn about that, but we'll focus our concentration on DNA. Now, what is this DNA? DNA is deoxyribonucleic acid. It's an abbreviated form. So DNA sounds pretty cool, right? Rather than deoxyribonucleic acid. So what is this DNA? DNA is the script for life. Now, let's see structurally what is it made up of. Then let's see the function of it. So DNA, as you say, you know, as I said, it's a biomolecule and it's made up of different types of monomers. Now, DNA has 4 types of monomers, 4 types of nitrogenous bases. The first one is called adenine, which is A, and the second one is guanine, which is G, and the third one is cytosine, which is C, and the fourth one is thymine. What our DNA is made up of? You can see, like, okay, our whole DNA is made up of just 4 letters, A, G, C, T. So DNA, not just AGCT, it's a stretch of this particular AGCT. It can be arranged in any random order, and there is a big molecule which is made up of only these 4 types of bases, that is A, G, C, and T. So what does this DNA do? Why is DNA so important? So DNA is nothing but it's a script of life, it's a blueprint of life, or it's the code of life. It contains the codes so that you become you. You are you because you have the human DNA. If you had the DNA of a banana, you would be a banana. If you had the DNA of a chimp, you would be a chimp. So that's what DNA does. DNA is so important because it has every information so that you can become a human being. So that's the power of DNA. So let's, to understand genetics, we need to know a little more about DNA. So how this DNA is organized, let's see that. So DNA, it's a stretch of nucleotides. The one here in the red color, the rope kind of structure, what you see, that's proteins called histones. They're octamers basically. So 8 histone molecules are there, and around it, these DNA is bound. And it is present. So here in this chart, you can see the DNA is there, it's wound around the histone proteins, and it is coiled into one level and coiled to the second level, coiled to the third level. Likewise, you have the supercoiled DNA, and the final supercoil, whatever you can see here, somewhat a scissor-shaped structure, that's called chromosome. This is just one form of appearance of DNA. Very tightly, compactly packed supercoiled DNA, that is your chromosomes. Now, humans have 23 pairs of chromosomes. The first 22 here, which you can see, those are called autosomes. The pair determines whether you are a man or a woman. So if you have a X and a X, you will be a woman. And if you have a X and a Y, you will be a man. So these are called sex chromosomes. So this determines the sex of an individual. So we know that now DNA is so compactly arranged inside the nucleus, inside a cell, right? So doesn't it amaze you when you think that only a stretch of nucleotides with AGCT, it's only 4 letters, which contains the information for life. So if you think about English as a language, how many letters it has? It has 26 alphabets. You have in Kannada somewhere around 45 to 50, and like all Indian languages have around so many alphabets. God, and it has 0 and 1. Likewise, our human information, the language of God, has 4 letters, that is AGCT. But how many of this is required to code the entire human being? It is 3 billion bases. So many bases, so many times AGCT, CTGA in a very random order. So many bases of AGCT or the DNA is required to code the entire human being. That many bases are required. Now, so this is too much. I said, when the DNA is supercoiled and arranged compactly inside a very tiny nucleus, the nucleus is less than 1 micrometer. It's like even less than 0.1 micrometer. That small nucleus, 3 billion bases are kept inside that tiny nucleus. Now, can anybody data how long it would be? Let's see that. So the y-axis here has the length in meters. if you can see, this is an average person. So average human being has a height of around 175 centimeters, 5'7", 5'8", somewhat. So it's around 1.75 meters, right? Does anybody know who is this?
No.
No? Okay, this is the tallest man ever lived on Earth, or ever recorded to be lived on Earth. And his name is Robert Waldo. He's an American. He was an American. And his height was 8.9 feet. So that comes around 2.72 DNA. DNA, if you take the DNA from a single cell and stretch it up, it's a little longer than the tallest man ever lived on Earth. It comes up to 3 meters. So it's this big, okay, from a single cell. Now, if you can imagine what will be the length of the DNA if you take like DNA from every cell of the human body. For that, we need to know how many cells human being has, right? It is this much. Can anybody tell me how much is this?
Trillion, right?
3 trillion. Trillion is the right. It is 30 trillions. Okay.
Sure.
Thank you. And that many number of cells we have inside our body, 30 trillion cells, 12 zeros after 3, right? So if you can imagine, if we take the DNA from every single nucleus of 30 trillion cells and stretch it up, how long it would be? The distance between Earth and Sun measures up to approximately 149 million kilometers. Okay, 149 million kilometers. And if you take the DNA from every single nucleus of every single cell from a single human body, that's 30 trillion cells, and stretch it up, you can to Earth 41 times. That's 6.2 billion kilometers. And this is all from a single human being. So we are so complex inside our body. We have 30 trillion cells and we have a DNA which can travel from sun to moon. You know, so complex body we have. And sometimes we, you know, complain about pimples or even some silly thing at work. He said that, she said this, gossips, all those things. I feel like laughing. See, you have such a long DNA and you have so many trillion cells inside you, which are working every minute. Right now, you're in front of your laptop or on your phone, watching this talk or hearing to this talk. And you're work and you're worrying about very slight imperfections in life. That's not fair, right? So moving on, as I said, DNA is a script of life. It contains code for a living being. So it's not just present inside the human being, it's present inside every living being. So DNA is inside a plant, it's inside an animal, it's inside even microbes. So if we take the DNA from a banana and DNA from a human being and match those two DNAs, we have a 50% match. You heard that, right? We share our DNA 50% with bananas. And if you take a fruit fly, we share 60%, 85%. And no wonder the chimp is laughing, because it shares 90%, 98% homology with the human being for its DNA. So actually, when the Human Genome Project, when the whole human genome was sequenced, it was done in 2003. Scientists thought that it was it. Okay, we know the DNA, we know life. But when they compared the DNA between these other organisms, they found a striking similarity. We and chimps have 98% similarity. That means, okay, me and chimp is only 2% different. Are we that much alike? So let's leave 98%, 85%, all this. Let's compare more. What about me and you? Different human beings. That is a 99.9% match. You and me have 99.9% match in the DNA content. That means me and, Sachin Tendulkar are 99.9% alike. Me and Lata Mangeshkar has 99.9% alike, similar DNA. But still we are so different, right? Okay, leave 99.9%, let's go to 100% and see what happens. So the picture you see here is identical twins, monozygotic twins, not the fraternal twins I'm talking about. They are identical twins, which means they have the same exact DNA. They are 100%. The match between their DNA is 100%. Identical twins, you can really say, even if leave that and consider a caterpillar and a butterfly. If you know the life cycle of a butterfly, these two are same, right? Egg into a caterpillar, and the caterpillar converts into a chrysalis, and then you have the butterfly, beautiful butterfly. But both of these have the same DNA. You have completely different looking caterpillar without wings and the beautiful butterfly with wings. But the DNA is an exact match, 100%. It's the different stages of the cycle, right? Leave the butterfly, compare the human being. When the human being in the womb, as a baby, as an adult, and as an old being, everywhere you have the same DNA. The DNA is permanent. DNA does not change with age. Okay, so DNA everywhere is same. But if I look back to my pictures like 10, 20 years old, I may not recognize myself. There is so much change, right? But DNA is not changing. DNA is the same. This made the scientists thinking, what is happening? Okay, if the DNA, even if the DNA has 100% match, there is so much difference which can't be excused with 0.1% or 2% or so. So the answer to this striking difference is the topic for today, that is called epigenetics. Now, epigenetics, epi, as Dr. Ramachandra sir rightly said, epi means, it's a Greek derived word, it means beyond, after, above, and genetics is the study of heredity. So this is something beyond genetics. Scientists discovered lately, in 2010, the Epigenome project has been started. After the Human Genome Project was completed in 2010, Human Epigenome Project has been started to understand the epigenetics. And it's been 10 years and it's still not complete. We do not know everything about epigenetics, but we know bits and bits of it, which I'm going to share in the coming slides. So epigenetics is something beyond genetics. It's an added layer of information beyond genetics, beyond DNA, which is making all those difference. So I said DNA is nothing but a sequence of AGCT. So if you consider this as 3 billion base pairs of DNA, which we have inside our body, not all of it is functional. Now, I can't say all of it is not functional, because some of it we do not know. We are yet to discover the functions of these 3 billion bases. We know only the function of few and the important ones, as we can say. So the one which are marked in red here are called genes. So these genes are the functional unit of the DNA. So gene, it can be converted. Gene is a code which can be converted into protein. And this protein, when expressed, does its function. So we are functioning. Okay. So there are stretches of nucleotides inside the DNA, which can be coded into genes. And not everything is coded into genes. The ones in greens, some of it we know the function, some of have the regulatory function, other functions, but the ones in red are genes which can code for proteins. So which can be, that means which can be expressed, which are the expressive part of the genome. Now, the difference between me and you is the level of expression of these genes. So I can have 100, like if you say a count is from 0 to 100, and have a 97% of some gene, you can have 96%. That will make a difference. And if I have a 100% of some gene and you have a 0.1%, 0.2% of some gene, that will make a huge difference, right? So the difference between me and you is the level of expression of genes and how these genes are regulated. Now how this happens? Okay, how does the genome know how much to express so that I can become me, you can become you? Now for that, there is a switch above every gene. So it's like the switch on and off button. So if the switch is on, the gene is expressed, and if the switch is off, the gene is not expressed. And it is present everywhere. And this switch is more like a regulator. It's not just on and off. It can be with different levels. You have the fan regulator where you can set the speed 0, 1, 2, 3, 4 like that. So similarly, it's like a regulator. It can have a different levels of expression varying from here to here. So this particular switch or regulator is the epigenetics. And this is making all the difference which we see between ourselves. So this is what epigenetics is. So if you compare the genetics or the DNA to a book or a script of life, epigenetics is like the highlighter, and which tells you which is important and which to quote, what to read in that book. And there are different, of course, there are different kinds of, different colors of the highlighter, which makes it more complex to read, right? And it's like having the same script, having the same almost similar DNA, having the same script and different movies. If you have seen these two, I don't know whether you have seen or not, The Great Gatsby in Hollywood, which was dubbed sort of in Hindi as Expose. One turned out to be a hit and one a flop. So with the same identical DNA, you can make a hit, you can make a flop. It's in the hands of epigenetics. So this is what converts the same script into different movies. So that's what epigenetics is. So epigenetics is, if you see the book definition, this is the heritable and acquired traits without altering the sequence of DNA. So let's understand a little technicality so that you can understand the examples which I'm going to give. So I said it's a switch. So switch is nothing but modifications on the DNA sequence. The DNA sequence itself is not altered. That is permanent for a lifetime. If you have a DNA, that same DNA is going to be there when you are born, and the same DNA is going to be there when you die. So DNA is not going to change. What is going to change is your epigenetics. So it's a slight chemical modifications on those AGCT nitrogenous bases, which tells the gene to switch on and off. One such, there are several hundred kinds of modifications which can happen. That's why the Human Genome Epigenome Project, which started in 2010, is still not complete because it's very complex. You have 100 different types of modifications at 100 different types of locations, and each one has different meaning. So we don't know how to read the God's language yet. So the DNA methylation, which I was talking about, one is addition of methyl groups, slight chemical molecules, which can be added just before where the gene is. And if the methyl group is there, the gene will not be expressed. It is switched off. It's like a switch off button. If the methyl group is there, the reader of the genome thinks it as, okay, this is off. I don't want to read this. And if the methyl group is not there, it is switched on. That means the reader reads this. Okay. So this is one example. And the second type of modification is histone modifications. So remember when I said about how the DNA is arranged so complexly inside the nucleus, we talked about one protein called histones around which this DNA is wound. And the modifications can also happen on these little proteins, histone proteins. And here the modification is not as direct as DNA modifications. It's little complex than that. You can have a methyl group added, or you can have other 100 groups added. It can be acetylation, it can be phosphorylation, it can be sumoylation, anything. So, and where it is added, whether it is added here, there is a meaning, and whether it is added here, there is another meaning. So reading histone modifications is very complex, but I just want you to know that this is also a kind of epigenetic modification. And a third kind of, epigenetic modification is microRNAs. This is where I am working on. I'm not going to talk about it much, but just an introduction. So you have a cell, you have a nucleus, you have the scribbled DNA here. So what if you have already the proteins, gene is already expressed, and the proteins are already in the cell. But after producing, you feel that, oh, okay, I did a mistake. Cell thinks that I did a mistake, I did not want that protein. What to do now? Epigenetics or the DNA modulation or the histone modification cannot do anything because the protein is already synthesized. How can you regulate that? Now, for that, you have microRNAs. So these are a type of RNAs which we omitted. Remember, we've omitted in the beginning. So this is a type of RNAs, it's very tiny RNA molecules, microRNAs. And what they can do is they can bind complementarily to the protein which is already synthesized and then stop the action of the protein. And hence you can regulate. So here, this is another level of regulation. So three types of epigenetic modifications we covered is DNA methylation, histone modification, different types of it, and another one is microRNAs. So we know how it happens, sort of. So now coming to epigenetics functionally, epigenetics is inherited and acquired. So some of the epigenetic epigenetic modifications can already be there in the gene, like some of the proteins which are always need to be expressed. For example, you have insulin. You might have heard about insulin protein, which plays a big role in diabetes, right? So insulin is a protein which is produced only in the pancreas. Now that means only pancreas cells should have should have a switch on button for insulin. None of the other body cells, none of the other body organs produce insulin. So all other body organs have a switch off button. All the body cells have a same DNA, but every cell is different, right? Inside the body itself, you have epigenetics, right? So all other body cells have a switch off button for insulin, which is kind of permanent. I can't say epigenetics is never permanent. It's persistent. Rather, you can use the word persistent. So all other body cells have a switch off button for the insulin gene, and only the pancreatic cells have the switch on button for the insulin gene. So these things are already from the epigenetic memory. We get it from our parents. So these things are persistent, okay? Some proteins which are always required, but some epigenetic modifications are acquired. That means this is how we interact with the environment. This is how we interact with the nature. So interacting with nature, we can change our epigenetics. So you can acquire happy epigenetics, epigenetic modifications, as well as you can have sad epigenetic modifications. It all depends on you, how you interact with the nature or the environment. And worst case, you can also acquire angry epigenetic modifications. So you don't want to go there, right? So I was speaking about inherited. So you have the sperm from the father, you have the mother, and egg from the mother. So both of the DNA have their own epigenetic modifications, right? Their own epigenetic levels. Now, when the child is born, I said, when I talked about chromosomes, I said humans have 23 pairs of chromosomes. We get the 23 chromosomes from father and the rest half from the mother. So these 23 pairs of chromosomes we have, and the two of them make up the child. And again, for the child, it can inherit some of the epigenetic modifications from the parents. And during the lifetime, as it ages, it interacts with the environment and gets or acquires its own epigenetic modifications. Okay. So this is what, inherited and acquired epigenetic modifications mean. And you might have heard this a lot. You are what you eat. Nowadays, people are becoming more health conscious. People are very much into like what you eat. You check the labels, you check the nutrition level, at least some of you, right? So you are what you eat. That's very true, right? But it is scientifically true as well. I'll tell you how. Now, as an example, I've taken honeybees. You know honeybees, right? Honeybees have 3 classes of bees. One is the queen. They live in a colony, and every colony has one queen, only one queen, and thousands of worker bees and few drone bees. Now, drone bees are male, worker and queen are female. Now, both, if you compare the genome of the queen and the worker, both of them have exactly same DNA. There is no difference, 100% match. But still, you have the queen, which is lean and with the shorter wings. It phenotypically, phenotypically it's different. And the worker bee, it has a longer wings because it does all the work, right? And you have inflated abdomen. So there is a difference between these 2 bees, but genetically they are same. What makes the difference? It is the epigenetics. So epigenetics of queen and worker is drastically, worker bee is drastically different. Now, what makes, how they are different? They are exactly in the similar environment. They live together, right? So the environment which they are faced with is similar. But what happens during the development? The queen is fed with royal jelly, something called royal jelly. It's a very highly nutritious food. And the diet of the worker bee is called worker jelly. It's a different composition. So the food which what they eat is different, and hence their epigenetics became different, and hence they are different. So this is a wonderful example for or wonderful illustration or a demonstration for you are what you eat, right? So this is not only true for honeybees, it's also true for humans. So you can look like this or you can look like this. It's all in your epigenetics. So how you can change your epigenetics? By eating healthy food and going for healthy choices. You eat your veggies, your epigenome will be very happy. It will say, okay, thank you for giving me this. I'll switch on the happy genes. And if you eat the fast food, junk food, which is not good for you, and you'll feel the epigenome inside that will feel, okay, why you are doing this to me? I'm going to switch on the sad genes. So that's what it does. And not only the food, it's also the exercise. There are plenty of studies which are done on these. And if you exercise, epigenome is going to think, okay, he's serious about it. I want to get lean. I want to get healthy. So your epigenetics will switch on the healthy genes and you can look like this. I'm not body shaming anybody. I'm not into fat and lean. I'm just speaking about the healthy and the unhealthy part. So if you're a little overweight and you're perfectly healthy, it's okay. But if you're, it has to be, it's a comparison between the healthy and the unhealthy. You don't want to be in this stage, right? So this is what the power of epigenetics. And that's one thing. You are what you eat. That's understandable. But the second level here is you're what your parents eat. Okay, you're what your parents eat. That's something unheard of. Okay, what my parents eat affects me. Is that a yes? It is yes. It is proven fact. So the picture, what you can see in the slide here, is mice. Epigenetic scientists love to work with mice and mice models. And the two mice which you can see here is the brown mice and brown mouse and the yellow mouse. These two are genetically identical. Just that the one with the yellow color is called agouti mice. It has an agouti gene switched on, and this is the normal mouse. So you have there is a gene inside a mouse called agouti gene. And if there is a methylation, the one with the brown color, it's lean, it's very healthy, and it lives a longer lifespan, and it does not get diseases that easily. So that is the normal brown mouse. It has the agouti gene switched off. While if you compare the agouti mice, it has the agouti gene switched on because there is no methylation on the DNA, and the genome reads it as switched on, and agouti gene is switched on. So this is a mouse with yellow color. It's very fat, and it is susceptible to so many metabolic disorders and different kinds of cardiovascular disorders, and it's not healthy, and it has a very less lifespan. So you are what your parents eat. For that, scientists did the experiment on agouti mice. They took agouti mouse and fed it with a very healthy diet on the left side, the one which you can see on the left side, and fed it with the no dietary supplementation and with the unhealthy food. So if a parent mouse was fed with healthy diet supplements, it had more healthy progeny and less of unhealthy progeny, or healthy baby mice. And if the mother mouse was fed with unhealthy diet, it had more of unhealthy mouse babies and less of healthy babies. So this illustrates you that what the parents eat affects the babies, right? Now I'm going to go further. What your grandparents eat. You will all be like, are you serious? My grandfather, somebody even don't know them. You know, grandfathers, grandparents are always cute, always helpful. Even now when I'm giving this talk, my mom is looking after my daughter, and they always come handy, right? They are so cute. Now, what they eat, the picture here is my father and my daughter. So what my father ate affects my daughter's health. Is it true? Seems it is. Scientists did the experiment with that too. And what they did was they fed one generation of mouse with very unhealthy diet. And they checked the generations, the babies, for 2 generations. And they found that the mouse were very unhealthy, and they were susceptible to metabolic disorders, cardiovascular disorders, and cancer as well. So you are what your grandparents eat as well. So these are all mouse models. You may ask me, do we have a human study? It's very difficult to do a human study because there are so many processes. You need to get ethical permission, and mainly you don't have that much genetical identity between people, and it's very difficult to compare the results. But there are few studies which already happened. I'm going to discuss a few with you. So there was a Dutch famine in 1944-45, soon after, at the end of Second World War, 1944-45. So in the eastern part of Holland, there was a scarcity of food. They were not allowing the transportation properly. So for 1 year, Dutch people, it's called hunger winter. The Dutch people suffered from scarcity of food. A normal human being, for them to work functionally, work properly, needs at least 2,000 to 2,500 calories per day. But during the Dutch famine, they only got at least like 500 calories per day. That was a severe scarcity of food which is ever recorded. So what scientists did was compare the babies or the generations which happened during this Dutch famine, like who were all pregnant. They studied their babies and their babies, okay? And they found that most of them had a very high susceptibility to diabetes and other cardiovascular disorders and cancer. So not only their babies, it's the babies, that is in the second generation as well, this was reflected. So it's very important to eat healthy when you are carrying, right? So because you can modify the epigenome of your baby. So that's about generations. Now, epigenetics is also related to your thoughts and emotions. That becomes little weird. You know, you take food, exercise, yoga is okay. Okay, that is modifying epigenetics, you can understand. But emotions, you know, it's not a tangible thing. Emotions, thoughts, it is known, it's a fact that they do influence your epigenetics and hence the overall performance of you. For this, they did a beautiful experiment on mice models. So what they did was, like when we have babies, what we do is we hug them in the initial periods, we hug, we kiss, and that's the one way of bonding with the baby, father and mother bonding with the baby. So that's very much required for the emotional support of the baby. But in mice models, in mouse, what happens is the mother mouse licks and grooms the baby for like 5 to 10 days. That's the way of bonding between the mouse baby and the mouse. So what they did was they had two groups. One group where the mother mouse was kept along with the baby, the licking and grooming was there. Basically the love was there. And in the second group, the mother was kept away from the mouse. And what they found was very interesting. And the baby, when grown up and became adult, the baby grown up to be like highly responsible to stress, and it was very healthy, emotionally stable, and performing all physical activities like a normal mouse. But the one without, which was deprived of its mother, which did not receive any licking and grooming, went on to be a very stressed, very furious mouse, and it was stressed very soon, and its stress response was nothing near to normal. So what happened, what is the basis of, molecular basis of this? If you see, you have a part called hippocampus in the brain, which is responsible for the emotional and social cognitive specialities. So inside brain, you have something called glucocorticoid receptors. These are like, this determines, if they are more, it determines you are more stable and you can handle stress beautifully. But they found that the one in the first group, mouse models in the first group had more expression of glucocorticoid receptors. That is because the epigenetics here was switched off. The methylation was switched off and they had more glucocorticoid receptors and the mouse was more happy. But in the second case, there were low glucocorticoid receptors and hence the mouse was furious and less responsive. So this is where you know epigenetics is influenced by thoughts and emotions as well, right? So epigenetics gives us two beautiful gifts for life. One is that you can give the sweet warning to your generations. If you are not eating healthy, exercising, not exercising properly, always with negative people. So that means it's a sweet warning to generations that you're going to suffer, right? For generations. And the second gift is regardless of what your parents ate, what your parents did, what your grandparents did, regardless of that, I said epigenetics is not permanent. It is tangible. You can modify it. So regardless of what they did, you still have the power to overcome it. You still have the power to be healthy. You still have the power to be happy. So that is the power of epigenetics. So if you ask me for advice, I'm not really good at giving advice all the time, but keeping this topic in mind, I might say go for an apple instead of a pizza, and go for a walk instead of lazily sitting at home watching Netflix, and spend time with your family, spend time with good people, positive people, spend time fruitfully. I can't say go out, travel, enjoy, because this is a COVID time. I can't give that advice, right? But spend time with them, spend quality time with them instead of gossiping or having negative thoughts, having negative emotions. Do the positive one. So the deeper we go, more we understand, but the more complex it gets. So we now know that we have a whole universe inside our body. How much complex it can be, right? And before ending, I would like to say DNA is not your destiny, but you are your destiny. So it's all in your hands. How you take your life, how you do your life. I would like to end the presentation with a very famous quote from Nelson Mandela. It always seems impossible until it's done. It's a very beautiful saying, because until somebody does it, you feel that it is impossible. Until somebody went to moon, we thought it was impossible. When somebody went, we now know that it's possible. So what I'm saying is that somebody can be you. Okay. So thank you very much for this opportunity. Now, when I'm saying thank you, my 30 trillion cells are saying thank you to you. Okay. So thank you very much for this opportunity. If you have any questions, you can ask me.
Thank you very much, madam, for this informative session. So if any participant wishes to interact with the speaker, or if you have any questions to clarify, you can post it in the chat box, or we can directly interact with the speaker.
So you can unmute yourself.
Hello. Hello.
Yes, sir, please.
I am Professor N. Gurnath Naidu, calling from Andhra Pradesh.
Yes.
Okay.
Madam,
Yes, sir.
you are nicely briefed about DNA and RNA and epigenetics. Well, I would like to just ask one question. Why people are so aggressive, in anger mode, highly emotional? As you said, 30 trillion cells are going to trigger for any activity. Why it is so high with some people and why it is so calm with some people? May I know the reason, please?
See, there are so many reasons. There are so many pathways, so many molecular mechanisms which can be active. So when I showed that agouti mouse model for fat mouse and the lean mouse, there was only one gene. But human beings are not like that. Human beings are very complex. You can't take one gene for one trait. You know, for aggressiveness, there is one gene. For height, there is one gene. It doesn't work like that. So there are plenty of genes at very different levels. And if it is at that particular level, your emotions are going to be like that. Now, what the inference of my talk was, it can be controlled with the food you eat and with the people you interact with, the like how stable you keep yourself. Hope I answered your question.
Thank you, madam. Thank you.
Anybody else want to speak with the speaker?
Hello, doctor.
Yes.
Hello.
Yes, you
sir.
can speak.
Hello.
Yes, sir.
Yes,
I
sir.
can hear you.
Please.
Yeah. Okay. Doctor, in a house or in a family, brothers, sisters, sister are at a matter of fact, the twins. They all grow in the same environment and they may have the same genes, but after their growth to adulthood, they change their behavior totally sometimes. Why it is done so?
Hello, can you hear me?
Yes, sir.
Please.
Yeah. So that again depends on the epigenetics. You know, yes, they may eat the same, but they too go to school, right? They interact with different people. They have different kinds of friends, and genetically they are not 100% identical. They have some difference, but again, how they interact with the environment makes that difference. How they perceive things, that also makes that difference.
Can I ask one more question, madam?
Yes, please. Yes, please.
See, very often we hear that diabetic gene has triggered. This has triggered, so that gentleman has become diabetes, right? Is it true or it's our behavior?
Diabetes is not a behavior, it's a diseased condition. Now,
It's a genetic.
Sorry?
It's a genetic makeup.
It is not always genetic. It can be a genetic phenomenon as well. So what happens is in diabetes, if you go to the molecular level, what happens is insulin, the type 2 diabetes, which is more common, you have insulin resistance. So what happens is your cells cannot take up what insulin does is to decrease the blood sugar level, and your cells cannot take up insulin, cannot use up insulin. They somehow become resistant to insulin. So it is not related to epigenetics. It's not triggered. It is a diseased condition. It's completely away from this topic.
Okay, so it is not triggering. It is our behavior in long range,
Yes,
the diet, exercise. That may
trigger in the
cause that.
sense. Sorry, triggering in the sense, say, if you have a long time unhealthy habits, it can be triggered.
Yes.
You know, beta cells, the pancreas, which produce the insulin, can stop producing insulin. So that can happen, but it is not as simple as it seems. It's not like if you eat this, this will happen. It's not as simple as that. It's a very complex network. There are multiple reasons for this.
Anyway, I compliment you for the beautiful and very easily understandable lecture, even for the non-genetic people. Good, thanks. Right.
Thank you so much, sir. Thank you so much.
So anybody else want to ask a few more questions to the speaker? Or you want to give your feedback, you can give. The speaker is here only.
Madam, now we are in COVID time, so still we are finding some medicine for this COVID virus. So is it related to DNA or RNA of the COVID virus that makes us impossible to find out exactly what exactly the medicine is?
That's actually a different aspect. See, viruses have the genetic material, which is very tangible, unlike humans. It is susceptible to change. They mutate very often. Humans, we have the DNA. As I said, when we are born, we have one DNA and we are dying with the same DNA. We don't change much. But virus, on the other hand, it keeps changing. It constantly mutates itself. So
it is called novel coronavirus. That's what. That's why it is called novel coronavirus. Every time it changes.
Yes. Yes. So when you are finding a vaccine, you are finding a protein in the coronavirus which can trigger our immune system, right? So scientists take up one protein and develop a vaccine, but by the time vaccine develops, the virus has already mutated and produces another type of protein. So that's why it becomes very difficult for a scientist to, get vaccines for viruses. Even for the common cold, we don't have vaccine yet, right? Effective vaccine.
Yeah.
So any more questions or you want to give feedback? So we have with us Shri Dinesh Kumar, the Secretary of Rotary Club Mysore-West, to say a few words. Anybody else you want to speak?
Shall we proceed, sir?
Yes.
Now I request today's guest, Shri Dinesh Kumar DK, the Secretary of Rotary Club of Mysore-West, to speak few words about today's webinar. Hello.
Yes, you are audible, sir.
Hello.
You are audible.
Yes, sir.
Yes, sir. Thank you very much, madam. On behalf of Rotary West, I congratulate you on excellent presentation, madam. Thank you very much.
Thank you so much, sir. Thank you.
So anybody else?
I think there are no more questions.
Yeah, you can.
I think there is a question in the chat box. You
Yes.
want me to answer? Is there a role for epigenetics in cancer? Yes, there is a very definite role of epigenetics in cancer. So cancer, what happens in cancer is you have uncontrolled division of cells. So for the cell to divide, so if you have a, for example, liver, you need to have 100 cells, but in cancer, you might have 200, 300, 500, like that cells. So it's uncontrolled, which is not needed. That division happens. So for the cell division to happen, there are many proteins which do that work. Now, it happens when it is not required. So by mistake, epigenetics thinks that it is required and it gets switched on, and the cells keep dividing, and you have multiple population of cells, which is not required. So this is how epigenetics plays the role there. So there are, lots of clinical trials are also in this range to stop that epigenetic modifications from happening, so that cancer cells, to stop the cancer cells from growing. Hope I have answered your question.
Okay, any more questions? Shall we proceed, sir?
Yes.
Okay, thank you very much, madam. I request Dr. Ramachandra C G, the Managing Trustee of SANATHANA SAMPRATHISHTANA, to summarize today's webinar. Over to you, sir.
Thank you, Prashanth. Today, let me once again thank today's speaker, Doctor, for accepting our invitation and to give more information about genetics. Even though we are also from science background, as she told, till 10th class to PUC we studied this biology, and it's an really it's an brush up session for us. She directly not started from genetics, all those things. She started from the base. She started with human body, what all the organs, so how the organs will perform its function, and she has given correlated this human body and organs to institution and different departments, how the different department works, then it will affect the reputation of the institution, like that. She calculated well example she has given. And the organs, so she said it is consists of a lot of tissues, and each and every tissue is performing its own function. So from the basic, she has given a lot of information, including the next part is cell. So that is the lowest, the functional unit of the body, or the organelles. So she told it might be like proteins, carbonates, all those things, information he has given. And nucleus information also she has given. It might be a DNA and RNA. She has taken DNA as a topic, and she explained more about DNA. That is, so far most of us DNA, so we know about it, but it is abbreviated one. So it's a deoxyribonucleic acid. So major constituent of this DNA is AGCT. So a number of AGCTs in a different the manner. So we'll be having in our DNA. And she also told DNA is a blueprint of our the body. And nucleosome model she has explained with a lot of examples. And chromosomes, she gave a very good example for chromosomes. So our human body will have 23 pairs of chromosomes, and the one XX, so if it matches, then female, and if it is XY, it is male. And DNA has got 3 billion bases, she told. And also we could not imagine the length of the DNA. She told the 30 trillion cells it contains, as well as 6.2 billion kilometer. So you cannot imagine the total length of our the DNA in our body. And a lot of examples she has given, how much percentage DNA it resembles, like a banana 50%, she told, honey is 60%, mice 85%, 98% is our land, 99% our human beings. So we differ from one another. And 100% twins babies, babies, she gave very good example. And she told, so from the day birth of a human being till he die, so DNA won't changes, it remains constant for one particular human being. And then she spoke about epigenetics. So genes, so it is nothing but a functional unit. And difference between human beings is the level of genes. That's what, even though 100% like twins are 99% we resembles, the DNA matches each other, but the level of gene will make a difference between one human being to the other human being. And genes which regulate, they are the regulators, like on and off switch everywhere. She told it very, and very good example also she has given how it works, everything. And stone modification, she spoke about it, how the modification will happen, like a euchromate, all those things she has given. And microRNAs she spoke about. So after that, so she told about epigenetic modification, how inherited the properties will going to get, even acquired the properties we get, so in our lifetime, all those things. And a lot of examples like parents baby, she has taken an example and beautifully narrated, so easily we could able to understand. And then she spoke about you are what you eat. So whatever we eat, okay, we get some sort of the habitats, like the honeybee example she gave, and human being. And also she gave very good example, so the best practices like food what we eat, definitely it reflects, and exercise and healthy, okay. So all those examples she has taken. The next one, she told you are what your parents eat. So this is very new to us. Okay, so whatever the parents so eat, okay, we are getting affected by that. So not only that, so that nice example she gave. And the third one is still it is beyond our expectation. You are what your grandparents ate. So even grandparents, what they would have went at that in their the cycle, it will affect us. So these are all some of the very new things we learned today. And, eat healthy food. That's what she gave the advice. And epigenetics, that means influence of the thoughts and emotions also. She told why the different people will behave in differently. And even so many participants have asked questions. Okay, even the siblings or the brothers sisters stay together, everything is same. So after growing up, okay, the entire their behavior and then everything will changes. So might be this, as rightly said by Adam, it is because of these things. And she gave nice examples there also. And at the end she narrated that this is the power of epigenetics, how we are different from each other. And a lot of good practices she gave advice. So definitely it is a eye-opening and a more informative session for us. Once again, I thank the speaker for sharing your knowledge to us. So what do you, Prashanth?
Thank you, sir. Gratitude is the essence of humanity. I thank Dr. Shruthi Kanthaje, Post-Doctoral Fellow, Yenepoya-Deemed to be University, Mangaluru, for accepting our request to be the resource person of today's webinar, and also for delivering a very informative session. Thank you very much, madam. I thank Dr. Ramachandra C G, Managing Trustee, SANATHANA SAMPRATHISHTANA, Dr. Raghavendra Prasad M D, President, and Shri Dinesh Kumar, Secretary, Rotary Club of Mysore-West, for their involvement in organizing this webinar. I also thank Shri Lokesh C G, President, Dr. Shantharaje Urs and Smt Bhagya Gopal, Board of Trustees, SANATHANA SAMPRATHISHTANA, for their support extended in organizing this webinar. Last but not the least, I thank all the participants for your continuous support and active participation in this webinar. Thank you one and all. Have a good day.